We're going to take a look at how myelinated axons propagate action potentials. So when myelinated axons propagate action potentials, that's called saltatory conduction. So the word saltatory means to leap or to jump. And that's just talking about how the action potentials are only going to be created at the nodes of Ranvier, these little gaps in the myelin sheath. And so it looks like the action potentials are leaping or jumping from node of Ranvier to node of Ranvier. So that's how they got the name saltatory conduction. And the myelinated axons are going to be conducting the action potentials a lot faster to the axon terminal than the non-myelinated axons. And we'll show you that with the animation here in just a little bit. And then don't forget, we've got some glial cells that are creating the myelin sheath. So in the central nervous system, do you remember which type of glial cell produces the myelin sheath? And hopefully you said it was the oligodendrocytes. So in the central nervous system, the oligodendrocytes create the myelin sheath. And then in the peripheral nervous system, which glial cells produce the myelin sheath? So you should have said the Schwann cells. So in the peripheral nervous system, it's the Schwann cells that produce the myelin sheath. All right, so let's see how the myelin sheath speeds up the conduction of the action potentials. So the myelin sheath actually starts after the axon hillock. So we'll reach threshold at the axon hillock. We'll open the voltage-gated sodium and potassium channels there. We'll create our action potential there. And then the sodium ions that are let in through the voltage-gated sodium channels would flow down through the axons, and then they'll hit this first node of Ranvier, and that'll cause those voltage-gated sodium and potassium channels to reach threshold, and that'll trigger the opening of the voltage-gated sodium channels. Sodium will rush in, and that'll create the depolarization phase of the action potential, and will create an action potential at that node of Ranvier. And then the sodium ions that have come into the cell will then flow down to the next node of Ranvier. And the myelin sheath is covering up the axon in this area in between the next node of Ranvier. So there's this myelin sheath right there. And the myelin sheath is acting like an insulator. So it prevents the sodium ions from escaping and being pumped out by the sodium potassium pumps. Because remember the myelin sheath? That's made out of the Schwann cells or the oligodendrocytes just wrapped around the axon, and so there's cell membrane there. And remember the sodium ions, they can't cross the cell membrane unless there's a transport protein. And so there's no transport proteins here, and so sodium can't get out. So it's just forced to flow down to the next node of Ranvier. So it just stays all concentrated here, and it doesn't dissipate at all. And so that allows this next node of Ranvier, where we've got a gap here, and then we've got our next grouping of voltage-gated sodium and potassium channels at this next node of Ranvier. So there's no voltage-gated sodium or potassium channels where the myelin sheath is, but they are located at these nodes of Ranvier. So at this next node of Ranvier, we reach threshold again. We trigger the opening of our next voltage-gated sodium and potassium channels. We create our next action potential We get more sodium ions coming in, and then they flow down to the next node of Ranvier, and then we'll open up our next set of voltage-gated sodium-potassium channels and create our next action potential. So we just create action potentials only at these nodes of Ranvier, because that's the only place where these voltage-gated sodium and potassium channels are. And that's really different from the axons that don't have a myelin sheath, because the ones that don't have a myelin sheath, you have to create an action potential all the way down the whole length of the axon. So at each place here, you've got to create an action potential, and that takes you a lot longer to get to the end where the axon terminal is. So the non-myelinated axons are going to be much slower in transmitting the action potentials to the end of the axon terminal. Okay, so here's just an animation that shows the transmission speed and the non-myelinated axon versus a myelinated axon. So we're going to watch the non-myelinated axon first, and notice how the action potential has to be recreated at each point all the way down the axon. So that's going to take a while until you reach the axon terminal. Okay, and then we've got the myelinated axon on the bottom here, and we only have to create our action potentials just at these nodes of Ranvier. So we're going to reach the axon terminal a lot quicker. So here we go with this one. So notice how much faster the action potentials are going to reach the end where the axon terminal is. So transmission speed of these myelinated axons is a lot faster. So we have two factors that are going to affect how fast the neuron is going to conduct the action potential. So we have the axon diameter, and then whether the axon is myelinated or not myelinated. So with the axon diameter, the larger the diameter, the faster the transmission speed. And if it's a smaller diameter axon, you're going to have more resistance to the sodium ions flowing through the axon, and so you'll have a slower transmission speed. So it's just like if you're drinking out of a glass with a straw, and if you have a larger diameter straw, you're going to have less resistance to the flow, and you're going to get more drink through your straw, and you'll have a faster transmission of your drink than if you had like a really small diameter straw that you're trying to drink your drink through, and you'd have a lot more resistance to flow, and so it would take you longer to drink your drink through that smaller diameter straw. So you'd have a slower transmission speed of your action potentials through your smaller diameter axon. Okay, and then obviously the myelinated axons are going to transmit a lot faster than the unmyelinated axons. So the fastest transmitting neurons in the body will be the large diameter myelinated axons. And then the slowest transmitting neurons in the body will be the small diameter unmyelinated axons. Okay, so we actually will classify the neurons based on the diameter size and whether they're myelinated or not. And they've actually named these. You don't have to memorize any of these, but we will be talking about these A delta fibers and these C fibers when we talk about pain later on in chapter 10. But the A alpha fibers, these are myelinated neurons, and they have the largest diameter. So they're going to have the fastest transmission speed they're going to be able to transmit the action potentials around 270 miles an hour. So that's really fast. And look who they are talking to, your skeletal muscle. And that makes sense because you want to be able to tell your skeletal muscle to contract and move and get out of the way, you know, if a car is coming at you or something. So that's pretty important. We also have fairly large diameter myelinated axons for some of our sensory neurons for like touch and pressure. And then looking at our C fibers here, these are not myelinated and they have the smallest diameter. So they're going to have the slowest transmission speed. So they transmit action potentials at about 2 miles an hour. So you can actually walk faster than they transmit their action potentials. Okay, so, and we'll talk about some of the pain signals that are transmitted this way. And there's actually 2 ways that pain is actually transmitted. There's these A delta fibers that do have myelin and they're a little bit larger diameter, so they do transmit pain signals faster than these C fibers. So there's actually 2 different types of pain signals we're going to learn about. There's fast pain and slow pain. And I know you've experienced this, and we'll talk about this later in chapter 10. So it's kind of interesting. Okay, and then this just kind of gives you some relative ideas of how fast these axons are transmitting. So here's your C fiber, really slow. They're not myelinated, very small diameter. You can walk faster though they transmit action potentials. And then here's these A delta fibers. They are myelinated, but and they're kind of a small diameter, but larger diameter than the C fibers. And these give you your fast pain. And then these A beta fibers, these are giving you some of your sensory, like touch and pressure. So they're pretty fast. So they're myelinated and have a fairly large diameter. And then just lastly, we've talked about this before. So when action potentials are happening and these voltage-gated sodium and potassium channels are opening, and sodium's coming in and potassium is leaving the neuron, there's actually only a small amount of sodium and potassium that are actually moving in and out of the cell. And so the sodium potassium pumps have a pretty easy time of putting the sodium and potassium back where they belong and maintaining those concentration gradients. So remember that the sodium potassium pump is going to be pumping the 3 sodium out of the cell and the 2 potassium into the cell. And that's what's going to keep the potassium high inside the cell, and it's also going to keep sodium high outside the cell and low inside. Okay, so make sure you memorize those concentration gradients for sodium and potassium. They're just the opposite of each other. And then also the sodium potassium pump is primary active transport. It directly uses energy. And then this is an important table from your book. There's a lot of potentials running around chapter 7. So make sure you know what the membrane potential is. And the resting membrane potential, know what a graded potential is, and an action potential, and the equilibrium potential for ions. So we've talked about all those. We'll talk about this receptor potential in chapter 10, so don't worry about that right now, okay? And we're not going to really talk about the synaptic potential, so don't worry about that, okay? And then I've got 2 really good animations here that go through the action potentials and talk about refractory periods. So they're fairly short and they would be good to watch at home and just kind of review the action potentials. So those are good to watch.